So welcome to video 5 in our short course on ST1 describing data. We're looking at sampling and experimental design, which is very, very important for your experiments. So we're looking at this process of inference. When we're taking a simple random sample, we're trying to infer population level data based on the samples that we get. So we're assuming that this distribution of samples we observe is representative of the true underlying population. However, that's the function of the number of samples that we collect. If we collect 5 samples, our estimated mean is one particular value. We'll find that our accuracy in terms of estimating the true population mean is very lower. It's because it's a function of the sample size. So if we have more samples, we'll find that our sampling error is lower. We get a better approximation of the true population mean, and then as our sample size becomes larger and larger. So this issue with the standard error will mean that more samples are always better because that sampling error is reduced. So our context for getting the sample is very, very obvious that we have a cost limitation as everything else, a time limitation. But we're assuming that our sample is representative of the population, and this is actually a major assumption, and it's quite hard to resolve this. A process by which we can extract samples randomly, as you already know, is simply to give each member of the population a number, use a random number generator to select some, e.g. using R code, and this is your sample. So remember that when we're allocating when we're doing sampling, what we're really thinking about if we have different groups is allocation. So we're trying to think about how we can incorporate randomization and indeed blinding into our experiments to prevent bias. So if we have 10 samples in total, we could take 5 random numbers between 1 and 10 and allocate pigs with those numbers to one group and the rest to the others. We want to use true random numbers, and that's a more nuanced area, but there are quasi-random numbers as well. But using R code is one simple way of achieving that. Issue with trueness in random numbers is sometimes to use the time and the clock to estimate the random number, which would mean that there could be correlations in the data over time if you're using a random number generator. So for instance, say you want to select 4 cows for testing from a sample of a herd of 30, and they have their 31. They have their names here. You might give them all a number and use R to generate numbers in this fashion, where they're using a runit function, which will generate 3 or 4 numbers, depending on what number you give it here, and they'll be between values of 0 and 30. So that's one simple way to generate random numbers. We also have to think about more nuanced experiments where you're dealing with a structure. So that might be, say, 2 categories, maybe infected and uninfected. So that we think about if their frequency of infected people is rare, how we access them. So for sampling from those different groups, the mix will never be exactly equal, and our sample will always be an approximation of the population. Randomly sampling people that happen or cows or whatever that happen to contain infected and uninfected will be unbiased and more accurate about the population level features. However, if we're dealing with a rare issue, this may limit our precision to actually understand the people who are affected or the feature of interest, especially if it is rare. So it's only going to be as accurate if we stratify them into groups, if the proportions in each group are exactly equal, which is never going to be that case. So this stratification means that we have to divide the population into groups, known as strata, and then select using simple random sampling or equivalent a random number of people. So that might mean we select if our frequencies of infected and uninfected are 20% and 80%, we might select 10 infected and 40 uninfected to give us a total of 50. However, there may be instances where we need a sufficient sample size inside the infected and uninfected groups, and this number of 10 will actually limit our inferential power here. Consequently, there may be studies where actually having 25 and 25, which is unrepresentative of the population, would actually be better. Indeed, depending on the study, they may want to oversample the infected and less have a fewer of the uninfected, because perhaps you understand the features of the uninfected less, and there's prior work on the uninfected has been already been completed. So sampling is not straightforward to do. There's also, so this is known as stratified random sampling, that we base on the frequencies and some proportionately. And then we're able to say, I put them into groups, grade 1, grade 2, and grade 3, and then sample from those different groups. One note when we're doing sampling is that this is actually done with replacement, which sounds unintuitive, but actually makes more sense. So if you have a music player that plays the same song twice in a row when it's on a random setting, then that is true randomness. And that is one nuance that we have to actually pay attention to, because true sampling contains replacement. If you sample without replacement, that is a slight bias. An example of that would be surveys that they're typically done without replacement. However, this is not usually an issue, because usually for very large populations, the probability of being sampled twice is low. So our main issue is that the standard error for might be very large for a particular group because the sample is very small. We may have other errors that come in here, technical errors and so on. Say if our freezer stop working, and that can affect a particular group of reagents, that can cause us major problems too. So we basically can look at different things that, if we think about the mean and standard deviation of our population being a bit like the accuracy and precision, we can get high accuracy if we stratified groups by sampling randomly across the whole group. However, if we want precision on a particular feature of interest within those groups, you want to compare the difference inside those groups, we have to look at precision, and we will lose accuracy. So a true random sample will give us good accuracy, but it may not tell us about the differences of interest. So the precision will allow reproducibility and tell us more about that. What we could do is hope that the feature has lower variance and increase the sample size, but that's not straightforward. So what we can do then is think about other sampling methods. So we might have many samples. So in this case, we might use systematic random sampling, where we essentially take every nth piece of data. So that might mean every 10th, 100th, 1,000th piece of data. So what we do, so say we want to estimate the mean expression of a gene, and we've lots and lots of images that we've grown in our lab. We've 900 of them. We can't sample them all. We can just deal with 20. We're going to look at RNA-seq 20. A way just do this is simply to label them all 1 to 900, generate a random number as the first number. So we might get 678 or something random showing up. And then we iterate, we take this out every 45th one after that. So go from 678 to 723 to 768 to 813, and so on and so on. And then we go back to the start of the list when we go past 900. So that's a way to randomly allocate them using systematic random sampling. Another sampling approach is known as cluster sampling. So here we divide the population into groups and randomly select all of some of those groups. So let's say we have a large set of cattle in a grassland. So we can think of their picture here. A bit like quadrants in ecology, we can just divide them into zones and say that, okay, we're going to look at, we've got 9 zones here, and we're going to look at all of the cattle in a random subset of three zones. So let's say we find out we have zones 2, and we have zone 6, and then we have zone 7. We're going to look at the cattle in those zones only. So that's a way of thinking about it. And it's another type of sampling, and gives us more depth about the features of the cattle within those zones. So it kind of depends on what question we're asking. Another example would be, let's say we have 10 antibiotic classes of interest, and each contains 10 compounds. So you got 100 in total, you're going to sample 50. So cluster sampling, in contrast to random sampling, would mean that we generate 5 random numbers for those classes, and we sample all the antibiotic compounds within those classes as part of our sample. And that gives us our 50. So we get more information about the variation within each of those classes, but maybe at a loss of precision about the broader features of the class level differences. So it's a kind of balance. But intra-group differences, we find out more at the loss of intergroup information. Our last major approach is known as convenience sample. So it's basically where you take, if you see social media with polls, that's an example of convenience sampling. They're not representative at all. They're just a random bunch of people who happen to be online or whatever at that particular point in time. Are they good? I don't know. But it could be an example would be interviewing customers or interviewing staff about their opinions on something. That might be an example of a convenience sample that it isn't necessarily representative or even to be interpreted in that way, but it is a sampling approach depending on what you're trying to look at. And if you're dealing with maybe areas of sociology, psychology, that's maybe where we're more relevant. For us, many of us are dealing with very, very small sample sizes. So this is where more complex designs can be useful. So one of those is known as kind of randomized block design, where we look at how we can partition the different variables across different features in experiments and understand better the main sources of error in our experiment. And when we talk about ANOVA in a course later, we'll look at that further. Another nice example is crossover studies where we have a limited number of, say, often we have a limited number of hosts that we can work with. That's very demanding. So think about how we can switch treatments at halfway. So let's say we have 3 equally good monoclonal antibody treatments for an infection. How can we use our limited number of hosts more effectively? So crossover treatments allow us to do that. So you might have, let's say here we have, let's say we only have 6 cattle, for example, and they're listed down here on this axis. So saying over time period 1, 2, and 3, the cattle 1 will undergo treatment A, then B, then C. And cattle number 2, cow number 2, will have treatment B, then C, then A. Then so on for this one, CBA, ACB, BAC, CBA. So what you note here, when we're switching from A to B, that can have effects, and B to C, that can have effects. And that's why the orders are not simply A, B, C, and A, B, C. They're A, C, B down here, as you can see, because we're going to look at the effects of A on C and B, C on B as well. So this is kind of each unit gets a treatment at different times. So each of these time periods, that's a variable in itself. And then we have the crossover effects as well. So not only we have the baseline, we have the treatment effect, we have the time effect, and the crossover effect to build into our model. So this is how we can use our data more effectively, but we also have to incorporate into that model, think about the sources of error. And what's great is that we can actually quantify these sources of error, take them away from our model, and actually measure the true effect of the treatment properly. So this can give more power usually. However, if A is really effective or B or C, then A, B, C, ACB, and so on will be less effective, and we might lose that information. So that is often where we're treating with, let's say, chronic infections or something where the treatment will help, but it's not going to be a cure. That's where this is most effective. And we also need to think about the carryover effect. So we're not putting in here is the need for more time inside these zones before we switch treatments. So these are known as washout periods. So with washout period, we'd have to probably do this over several trials, but basically measure at the end of treatment A, but also measure at the washout period where we expect some small effects maybe from A, we're not sure, and then at the end of B and the end of this washout period as well. So expect the effect of A to be lasting during this A period. So that might extend beyond that treatment. If we think it still has effects, then the washout should have no effect. That's the assumption, and that's a quantifiable assumption. Does it have no effect? Same for B and C and so on. So in many senses here, we're actually dealing with 6 discrete time periods here. So this will help us control for false positives. We can think about further design. So Latin squares block design is a crossover design such that each treatment is used at least once per sample. So if you think about if we have 4 treatments, A, B, C, D, we can have a treatment going from A into B into C into D, but also we can have just reverse them, A here at the end, then B, C, D, then C, D, A, then D, A, B, C. Of course, we can rearrange these further such that we're avoiding only measuring the effects of A crossing over into B and B into C and C into D. So that's actually a quantifiable component as well to maximize the number of independent crossover events. So sufficient power to look at the effect of A on B, but also A on C, which is not measured here. If we have 2 treatments and 2 samples, this is pretty easy to do. We just have 2 factorial treatment sequences, A, B, and B, A. So that's easy to do. And if we have 3 treatments, we have 3 factorial possible sequences here. Here we have four factorial treatments, but this is just one of 24 options. What is good about Latin squares block design is that we can change the treatment periods to make sure that the design is balanced. So here we have A is treated A crossing over into B, A crossing over into C, A crossing over into D, and then A into A. So you see there's an extra time period here to control for the effect of D moving on to B. So that means that we have all the different effects of A affecting not only itself, but B, C, and D as well. So this is a balanced block design. So we have 4 groups here, and then 5 time periods, ignoring the washout periods. So we can say that this is a strongly balanced design because A is preceded by A as well as all the other options. So when we think about the models associated with this, so when we think about this in A, there's several features that we can think about. So baseline effect, there could be a carryover effect, there's the sequence effect, and the time period. So we can quantify this. So we can think about we're trying to measure some gene X. So it depends on the observed value, the treatment effect, but the carryover effect as well. In the baseline level, the expression might be non-zero, and then there might be effects from the time period. When we're doing experiments, there could be effects from the temperature, the pressure, other things, kind of small effects, and they're quantifiable. Hopefully they're 0, but we can think about them in more detail. So if we think of a more simpler example where we just have A and B here, we can think that for A and B here, we just have to measure a few different things. We just have to measure the mean of A. That's mu. I times that mu. Mean of A. Then we have the sequence effect. So we've got the sequence effect. We've got a carryover effect, which is 0 for this A here at least, because there's none. And then there's the time period. So it's time period 1. So that's the variables in the first one. And then for B, it starts at mu for B. The sequence effect this time for B and A. The carryover effect is going to be 0. And again, we're looking at time period 1. And what we should think that is that our attempt to estimate mu of A is simply based on the observations we have here. So that's going to be based on this cell and this cell. So that means our estimate, the mu of A, is going to be based on values here and for this other cell and that other cell. If we change our values here, we're going to have treatment 2, we're going to have some sort of carryover effect from B. We've got the sequence effect of A, and mu of A. So that means these carryover effect and the time effect can actually affect our estimate of mu. So if we write this down here, so in cell A1 here, so A1 is over here. This is A1. That's a 1 here. This is B1, B2, and A2 is here. So the mean for A is going to be this and this. So you can see there we're taking out the carryover here because there's none. And same for this, we're taking out the carryover. So if we calculate the mean effect of A, it's going to be A1 plus A2. That's in terms of what we detect. So you can see that if we do that, then we actually simplify that. We got mu of A plus the carrier effect of B divided by 2. So whatever difference is the time periods will cancel out because we only have 2 time periods. So averaging them out should not make a difference. And then we get the carrier effect. So our estimated value, our expected value for the mean effect of A is going to go from the mu of A plus the carrier effect of B. So if the carryover effect is large, that will affect our estimate of the true mean. It'll be skewed. If the carryover is very small, then we're fine. So this means it's hard to estimate if the carrier effect is very large. So if we think about this in terms of real studies, we can look further and look at how they've done this. So these are more simple examples. So if we have a standard vaccine studies allocate a vaccine group and a placebo group, and what we have in this particular study, taken from this paper, is an example where they monitor the vaccine group and the placebo group using a trial sample size to see if there's early efficacy or not indeed. And what they do then is that they give the vaccine to the placebo group once that is established. So once there's sufficient evidence of placebo group, so then what we actually deal with then is an inferred imaginary placebo group, because maybe it's not ethically wise to avoid giving them the vaccine at this point because early efficacy has been established. And then we can still monitor the effects in the vaccine group and this new vaccine group to compare it to the data we had for the placebo group to infer the estimated vaccine efficiency. So there's an example here. We're looking at placebo and vaccine groups. This is our vaccine group and placebo groups. You can see there's actually 20 people across here. So you got 14 total on just one instance here. So that's 1 out of 40 for a vaccine group. And then for placebo group, you got 1 there, 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 and there. So you got 5 out of 40. So we can compare those relative rates to estimate the vaccine efficacy. So it's simply 1 out of 40 divided by 5 out of 40, and you take 1 away from that value to get the fraction that of denoting the efficacy. So if we look at that, efficacy here, we'd get a fraction of 80%. 80% efficacy. So if you look at the original vaccine group, so 1 person dies, you got 5 out of 39 here for this group. And then for their deferred vaccine group, we got 2 out of 35. So that means we can use these two values to compare against the inferred placebo group. So that means we can get the inferred efficacy for period 2 of being slightly lower at 67%. So there's other factors going on here. So obviously, this group for period 2, this group may have been vaccinated here at this stage at the start, and this is a later stage suggesting that the vaccine might have reduced effect with time, and that'd be something that could be monitored then in subsequent periods. An example from this paper is known as ring vaccination, which is quite a different design compared to crossover. So for the oral cholera vaccine, what they use is when they detect a case, they inoculate the people with the vaccine who are maybe in contact with a case, and then monitor the outcomes to see how effective that vaccination is. And that's different to the random vaccination, where we're assuming that a certain fraction of the population will come forward to be vaccinated. So that's a slightly different design, but it's used widely for certain rare disease. So the Nipah virus is an example of that. Some trials use this kind of vaccination where they vaccinate immediate contacts, but then also delay vaccination in others to see if there's a difference in terms of the efficacy of that vaccine. Other studies have used cluster-based approaches where they vaccinate extensively one group, but not another group to see, again, if the vaccine is effective. And then there's a classic mass vaccination as well, where there's also potential to stratify them based on geographic splitting. Another nuance with sampling is to think about the efficacy of treatments. So we might be looking at antibody treatment, and we can think about these special terms: equivalence and non-inferiority. So non-inferiority indicates that some new treatment does it have acceptably lower efficacy compared to the existing treatment. So this might be for good reasons. So treatment B might be cheaper or easier to administer. And we have to consider studies where it's not ethically possible to have a complete placebo group. We have to have treatments for both groups. So we can think about non-inferiority as being acceptably lower. Equivalence is being the same. So think about drug manufacturers obviously have to go through a lot of extensive testing to see if their batches perform in a way that is the same as existing batches. So if we're thinking about the treatment difference, we could have an example of a trial where the confidence interval and mainly the point estimate can show a definite drop compared to an existing treatment. Non-inferiority might mean that our estimate of the new treatment B could be lower than the existing treatment, but it might be comparably the same. And then equivalence would mean that the evidence would suggest that there's no difference between the treatments. So this is kind of a difference of where the point estimate lies here in the middle and how our confidence or credibility interval overlaps that of the original trial. So an example of this would be childhood asthma, where we need a placebo. Having a placebo would not be wise. So this detailed more from that paper is detailed in with more detail here. Typically, we use comparative studies where we have some new treatment and some old treatment, and we see if is there a big difference. It works both ways. We want to see if the treatment is better or worse. These are our typical studies we're used to. And then we have our graph for equivalence studies where we have equivalence, a lower effect, or indeed a higher effect. So it's not actually the same. Non-inferiority compares then is it within the same bounds as our existing treatment in effect. So this means you have to think carefully of the differences between, say, a clinical controlled trial, which looks at the precise effect of a subgroup versus a randomized controlled trial, which simply looks at the whole sample that we have and allocates them to particular features randomly. These are not the same thing, and they will tell us different types of information. So though randomized controlled trials can be used for many different things, they aren't necessarily the best to be used in certain situations. So this goes back to a nice paper by Leek and Peng that you should look up. It was in the Science magazine, which simply asks, what is the question? So key steps in analyzing biological data are simply to summarize it, number one, and that's what we're going to look at later. Did you summarize it? That gives you data to look at. Did you report the summaries without interpretation? Well, that's number one, descriptive. a lot of the work that we do is simply descriptive. It's simply to understand what do we have here, what does it look like. Then you can move on to the next stage and ask, in new samples, do your descriptions, basic information, does that hold? Is it the same or is it different? And why if not? You can extend this further then going beyond exploratory analysis, but if you think about it carefully, are you using a model to compare how one variable affects another variable? If no, are you predicting for individuals or for groups? Most of the time, we'll be predicting for groups. It's very seldom that we'll actually be using prediction in the technical description of the term. So actually, we think when we're using scientific language, we should be careful when we use the term predict, because that actually means a lot. Predicting for individuals is very, very hard. Making inferences for populations based on sample data, that is more possible, and most of the time we're doing that. So building up here from describing to exploring in a new cohort to inferring overall features. And maybe if we do that, we could possibly make predictions for individuals, but I think that's quite a hard problem to solve. Then we could look at average effects in the terms of causal models, or indeed mechanistic effects. But you're probably thinking of these in terms of more in functional studies that probably have to be tied here to show true causality or mechanistic effects in biological terms. So we can ask ourselves to help us define what problem we're trying to ask, and that will help us better design studies. So what type of data is being examined? Have you developed a model? It does work for this data, and why this model? If your hypothesis is correct, then what follows from that? And even if your hypothesis is correct, what are the boundary conditions for that? What would mean that it wouldn't work or wouldn't work to a suitable, acceptable standard? So which model is actually a big question. And if you think about our assumptions about different models, often with biological data is no inherent model. But a lot of statistical methods assume models. And we think about parametric methods and non-parametric methods. Non-parametric methods can use ranks and other comparable information. But often our assumptions about parametric methods assume that data is normally distributed, which doesn't actually work. But our data may conform to another model. It could be Pareto, it could be Weibull, it could be left-right centered, it could be hypergeometric, half a T distribution. This is by Bath. It could be just a normal distribution, in which case parametric works, but it could be binomial, as we had the examples of coin tossing here. It could be a gamma distribution or a Poisson distribution if we're looking at events. It could be an F distribution if we're looking at ANOVA or chi-square tests. There are lots of options. So to think about this carefully for biology, if we think about what is a null phylogeny, what do we expect to get from draw phylogeny? Is there any real null here? If we're looking at gene expression, what is our null value there? What does it look like? And if we're looking at cell types, what is our typical cell type? What's cell type? What's our average cell type? Is there an average cell type? Does that have any meaning here? If we want to convert our biological phenomena into statistical models, careful thought is needed. And that's why I warned that I'm in the category of people that think having purely hypothesis-driven research is dangerous and misleading, and it leads to self-reinforcing research where everyone assumes that P53 or FOXP2 or your candidate gene causes everything. That has been shown to be very, very misleading. So if you think of this at the level of complex processes, whether molecular, ecological, or otherwise, and not just particular molecules that do a lot of things. Our problem is simply that our causality stems from the description of our data and our study, and that stems from the design of our study, and that feeds into causality. So you end up just in a circle. These feed back into each other, they bleed over onto each other, and we get causal inference based on actually bad design and bad analysis. And I can't tell you how many papers I've rejected as an editor because they've analyzed their data with Graph Prism or some other software that does not allow them to describe their data in true scientific terms. So we have to really think carefully. This is from McClure et al. Looking at our understanding and our scientific papers and how analytical approaches that we might use might affect our study, how we might misclassify data, we might select different data sets or methods or machines or whatever. That might bias our data. We might have confounders here where we're assuming X causes B when X is we're assuming X causes Z, whereas there's another variable Y that's causing both X and Z. And we think about random error and how that can affect our experiments too, and how that can mask all of these can mask the causal effects. So some questions you can look at for this whole area include, say you imagine you have a big freezer full of infected and uninfected chicken thymus tissues. If you look at protein A in a study using 20 samples allocated using random numbers, and study B looks at protein A again using 10 infected samples and 10 uninfected samples, which is more accurate here and why? And there are these questions of sampling. We're looking at a study looking at the number of bacteria per petri dish in a big collection of 10 species. What is the type of sampling in each of these cases? So you take the 100 petri dishes by selecting 10 for each of 10 species. Number two, you use a random number generator to select a petri dish with the numerical IDs of all the petri dishes. Starting with that petri dish, every 40th plate is chosen until the 100 plates are in the sample. Three, a completely random method is used to select 100 petri dishes. Each petri dish has the same chance. The species are number 1 to 10. Remember, we had 10 species at the start. And a random number generator is used to pick 2 of these species. All the plates in these 2 species are in the sample. And then finally, number 5, you're asked to grab the first 100 plates that you find in the freezer because we need the results ASAP. Okay, another question. You've got 3 test monoclonal antibiotic treatments and only 9 cows that you can inoculate with the virus that they treat. So how would you design the experimental setup to maximize your power? And you can, you think about costs, think about time, there's lots of variables, facilities. All of these kind of impact how we approach these things. Extending from question 7, now describe this as a mathematical model denoting realistic potential variables that would affect your data. And say your variable of interest is your viremia levels in these cows that you have. And then going back to vaccine efficacy, you can infer, then deduce, what results would you need to obtain a vaccine efficacy of 70% and inferred vaccine efficacy of 85%. Obviously, there are many options for calculating the underlying data for this, but any one is okay. Finally, then when the term predict is used in statistics, what are the requirements for the input data and prior results are needed for that idea prediction? And question 11 is transform your data beyond using log-based transformation and exponential-based transformation. Is that permitted in research? So that's the end of this video. Please look at the next video on data summaries for more information.